All right, guys, as we move forward with antibiotics, it's really important that we have some basic terminology down going onward here. So we're going to just introduce those basic terms. All right, let's jump right into it. Okay, so when we talk about antibiotics, we will refer to natural antibiotics at times. These are chemicals that are produced by microbes. All right, we have to keep in mind that microbes, many of them, the bacteria we talk about, contain these plasmids. These plasmids, some of them are bacteriocin or antibiotic plasmids. They have genes that code for chemicals, which they will protect themselves with and fight for nutrients and space against other microbes, right? So we need to keep in mind that antibiotics did not start with some researcher in the lab, just kind of, thinking up chemicals we can make to kill. These specific killers actually were made by microbes, and we're just stealing this idea most of the time, okay? So natural antibiotics are going to be produced by microbes themselves. In fact, we talked about how it was that bacillus organisms and different fungi and actinomycetes from the soil all are capable of killing when it comes to antibiotics they produce. Okay. We could also talk about synthetic antibiotics. Okay. These would not naturally be synthesized by microbes, but typically we are looking at the chemical and the chemical structure that is produced by the microbes, and then chemists in the laboratory will go about reproducing that chemical through chemical pathways that they understand, right? So synthetics are completely made in the laboratory. However, again, we're stealing the idea by looking at natural antibiotics and then trying to reproduce them in the lab. Finally, we can talk about semi-synthetic antibiotics. These semi-synthetic drugs would be derivatives of the natural antibiotics. So in this case, you're taking natural antibiotics, naturally occurring chemicals, collecting them and purifying them, and then altering them chemically in the laboratory to achieve certain characteristics that might be beneficial. Many natural antibiotics have some pretty nasty side effects in the animal system, in the human body, okay? And so oftentimes we're trying to alter those naturally occurring chemicals so that they're not quite as harsh or don't stimulate the same kind of rapid immune response or whatever, or have allergies or whatever that is, okay? All right, so we have natural, synthetics, and semi-synthetics. This idea of semi-synthetics is not at all new. I'll give you an example. It's not an antibiotic drug, but Dr. Bain, who happens to be a chemistry professor over at BU, at Binghamton University, she's been working with Taxol derivatives for some time. She used to take Taxol is actually a drug that is used against cancer, and it has an effect on cell division. And what she would do is take this Taxol, which is a naturally occurring chemical from, I believe, the bark of yew trees, which you can find in the tropics, and she would then alter those chemicals to produce slightly different variations of Taxol with the hope that it has some beneficial characteristics that would be useful in industry and pharmaceuticals, okay? So it's not at all uncommon with other kinds of drugs to make semi-synthetics. All right, another one, and this is somewhat intuitive, I think, therapeutic versus toxic. So a toxic dose, do you want that? Sounds like pretty much a no-brainer, right? I don't want a toxic dose that would be toxic to me, right? I want therapy. I want something that would be therapeutic and would help me, right? So a therapeutic dose would be the quantity of a drug that drug level, right, which would be required for the clinical treatment of an infection, right? So if I have a bacterial infection, I want the amount that's going to be effective and therapeutic without hurting me, right? Without doing harm to me, to my cells, versus a toxic dose. Now, a toxic dose, typically a more concentrated, a higher dose, right? A drug level that's higher than a therapeutic dose. Of course, it would kill the bacterium, most likely, right? In the laboratory. But what makes it toxic is what it does to the host, right? To the host that has the infection. So we don't want a toxic dose. We want a drug that's effective at a low enough concentration in the body that it won't cause harm to that host, to that patient, right? So this one's kind of an intuitive one, kind of a no-brainer when it comes to that. All right, we need to concern ourselves with spectrum, the spectrum of activity that a drug has or is capable of, all right? So here we'd be talking about narrow spectrum versus broad spectrum activity of a drug. If a drug has a narrow spectrum, it will be effective against a limited variety of pathogens. So let's say we're just talking about bacteria. We could classify bacteria by doing a gram stain, right? So let's say we jump in, we do a gram stain. Is it gram positive? Is it gram negative? We've got a few, right, that might not even have a wall. So what this is saying is that some drugs are effective against, let's say, just gram positives, but all the rest, they could care less, right? Or this is just effective against gram negatives. That would be more narrow in that it's just picking one group, right? And you could get even more narrow than that, because within gram-negative organisms, there are lots of other subclassifications, right? So you could imagine one gram negative that's susceptible to a drug we use, and the other one could care less, right? It just keeps on growing. Broad spectrum would be just what it sounds like, a more broad killing of cells, right, of bacteria. So if I have a broad spectrum activity, it might kill gram positive and gram negative. Or let's use a different classification. Let's talk about, is it aerobic or anaerobic? A drug could kill just aerobic organisms, narrow, right? Or it could be more broad. Maybe it kills anaerobic and aerobic organisms. So it doesn't have to be cell wall related. It could be related to other characteristics. This is why, guys, that we spent all of this time trying to differentiate these cells from eukaryotes and from each other. That's all it was about, right? And especially in medical field, in clinical terms, this is what it's about, right? All right, let's think about, is the drug bactericidal? Is it bactericidal? Like, I hear cidal, I think, right? Other terms like homicide or suicide, right? We're talking about killing, right? If a life is lost, and in this case we're looking at bacteria, bactericidal would kill the cell, right? It would kill the susceptible bacterium. Unless, if it's resistant, obviously it wouldn't work. But susceptible is why that word is there, right? So this bacterium has no defense, it would kill it. This drug would kill it. Versus a bacteriostatic antimicrobial or antibiotic, it would reversibly inhibit the growth. Reversibly. I'm not outright killing the cell. I'm just adding a drug that will stop the growth. It will stop the cell from going through binary fission. The population won't grow in size. You might think, well, what's the use? Why wouldn't you use bactericidal? Remember, we can cause some harm, especially with like gram negatives, if we blast that outer membrane apart. LPS molecules go everywhere and we have anaphylactic shock. So our goal may be, in fact, to pause the growth of that infection, give our immune system a chance, right, to get ahead of this infection and to kill those organisms. All right, another term, another concept we need to know is what is combination therapy when it comes to antibiotics. A combination of different drugs being used. And here too, it's important to understand why we would use a combination. The definition of this is fairly obvious and intuitive, right? I use a combination of drugs, not just one, right? So maybe I'll use antibiotic A and antibiotic B together, a very specific choice of the two I want to have the effect, to be therapeutic. So why would I do this? What are the reasons that a doctor might order up two drugs rather than one? One is that it's possible that the use of two or more antibiotics would be helpful in that it would prevent the emergence of resistant strains. Okay, so if I use one drug that will kill most of the population causing the infection, but there's some small subset of those cells that are resistant because they have a resistance plasmid, well, I might want to add this second drug that kills in a different way so that those that are susceptible to antibiotic A are going to be killed also, right? So that a separate population of the cells causing infection, perhaps the same species, but some that have resistance don't cause the infection to be worse, right? So that could be one reason. Another reason may be that I haven't gotten my results back on the sputum sample that I took. When it comes to, maybe you think it's strep throat, but you're not sure. Maybe you need to wait for the organism to be cultured, and that can take a day or two. Now, some tests are much quicker, right? But that isn't true for all cells. Sometimes we have to grow it first to know what the best next step would be. So if you don't want to wait for that, you may want to apply a couple different drugs, taking your best guess that it's or either bacterium A or bacterium B that are causing this infection. So I'm going to choose antibiotic A and antibiotic B to cover both bases. And finally, it's possible that choosing two different drugs will make them better together. All right, so we're gonna address that on the next slide. What is synergy? What is a synergism? Okay, it's basically a situation where both antibiotics work to a certain level, but when I put them together, they work even better. It's going to multiply the effectiveness. It's not an additive kind of thing. It even makes it better than that. All right. So here is the definition of an antibiotic synergism, a synergy. Okay. The effects of a combination of antibiotics would be greater than the sum of the effects of the antibiotics individually, right? So if antibiotic A kills X number of bacteria and antibiotic B kills Y number of bacteria, it's not X plus Y. Okay? The synergy would give you greater than the X plus Y effectiveness. All right? And we'll see with a later clip how it is that we can actually test whether two drugs have a synergy in a laboratory. Okay? It's also true, though, that drugs could antagonize one another, right? When you antagonize your little sibling, right, you kind of just bug them until they explode, right? So you're kind of inhibiting their ability to do whatever it is they want to do, right? You're kind of bothering them. So here, when you antagonize, when one drug has an antagonism with another, it's going to lessen the effectiveness. It's the opposite of a synergy, where it multiplies the effectiveness, right? So this occurs when one antibiotic, usually the one with least effect, interferes with the effects of another antibiotic, right? So it's going to lessen the ability to treat that infection. All right, now, it will be critical you have the definitions of these down, all right? I'm going to go over some drugs very soon with you, the different types of antibiotics that we work with, and the categories of those types. But there's lots and lots of drug names, all right? I won't necessarily expect you to know, if I throw a drug name at you, that you have to know, is it narrow spectrum, is it broad spectrum? Does it have synergies with other drugs, or does it have an antagonism against other drugs? These are all things that are documented, and they're in databases, and we can look those things up. And if you're in the clinical setting, you'll probably get to know them, right? You'll have them, you can come up with them pretty quickly. But in this test, at least having the definition is critical. Okay, so if I tell you that a particular drug, and on the test I say that this particular drug has a synergy with another, or flip it, I could say drug A and drug B work even better together. What does this sound like? You'd want to know that sounds like a synergy, right? Based on your definition, your knowledge of that definition. Okay? So I'm going to leave it at that, and we'll jump right into how is it that we test for the concentrations we need in the laboratory. How do we get started researching these drugs? All right?